right. Thanks for coming out, guys. So we're going to travel back to the Mesoarchean today, but before we do, I wanted to thank my funding sources, which is the current NAI CAN cycle, the Lewis and Clark grant, and the GSA Graduate Research Grant. And I also like to thank my collaborators. So I'm going to start off today talking about the motivation behind my research. Then I'm going to go into background into distillatory iron reduction. I'm then going to go into the approach behind my research and give some background to my field area. And then I'll present some iron isotope data, put it into context in a model, and tie everything together with some implications and conclusions. So the question driving my research is, was there a chemical footprint of distillatory iron reduction 3 billion years ago? And I ask this question because distillatory iron reduction has great astrobiological potential. Its electron acceptor iron is widespread in the solar system, and this metabolism of distillatory iron reduction is deeply rooted in the tree of life. So let's move on to some background. This equation right here is showing distillatory iron reduction with the electron acceptor in blue and the electron donor in red. So in this process, you oxidize the organic matter at the same time that you reduce the ferric iron, and this produces ferrous iron. The purple boxes here are indicating the phyla that are capable of distillatory iron reduction. So you can see that those phyla are deeply rooted, and they're present in both the bacterial and archaeal branches of life. So how do we find DIR in the rock record? What fingerprints are there? It basically boils down to isotopes and minerals. So if you guys aren't familiar with iron isotopes, we've got the notation here. So iron isotopes are measured relative to a standard, which is the average igneous rocks. Iron 56 is the heavy isotope of iron, and iron 54 is the light isotope. So if you get a positive delta 56 iron value, that means you're enriched in the heavy isotope relative to igneous rocks. And if you get a negative value, that means you're depleted in the heavy isotope. Now this process, it also enables the formation of certain minerals, such as magnetite, siderite, and vivianite. So combining isotopes and minerals, we can find DIR in the rock record. So for my study, I did iron isotopes on whole rock samples, microgyl pyrite, and magnetite separates. These samples come from the Mesoarchean Witwatersrand supergroup, and they represent a wide range of lithologies and depositional facies. So where is the Witwatersrand? The Witwatersrand supergroup is part of the Katval Craton of South Africa, and it's composed of the older West Rand group and the younger, more famous Central Rand group. And the Central Rand group is very famous because it has lots of gold. But my samples are from the West Rand group. And we are lucky to get those samples because when they were trying to sample the Central Rand group, sometimes the drill cores went down too far and they accidentally sampled the West Rand group. So I'm very thankful for that. So there's a couple really fantastic aspects of the West Rand group. The first is that it's experienced low greenschist facies metamorphism, which sounds pretty scary to anyone that works with younger rocks or modern sediments, but for the Mesoarchean, it's pretty much the best we can hope for. These rocks represent a complete depositional basin from just offshore to the starved shelf. And these rocks have been used as evidence for atmospheric oxygen. And that has come in the form of chromium isotopes, selenium concentrations in pyrite, and molybdenum isotopes. So I say that we have this whole complete depositional basin. Where did I sample from this basin? So you can see here that this is a sort of model ocean basin. And so we have samples from the braided fluvial depot facies, which is just offshore, and then slightly inshore, the pro-delta depot facies. And these would be considered proximal. The transitional shelf would be intermediate, and the outer shelf and starved shelf would be the distal depositional facies. So let's look at some data. This is showing iron isotopes plotted against total iron concentrations, which is normalized to aluminum. And we normalize to aluminum because we want to look at iron enrichment beyond detrital flux. So the cooler blue colors are more proximal depositional facies, and the more red, warmer colors are the more distal depositional facies. The diamonds are microgill pyrite, the circles are whole rocks, and the squares are magnetite separates. So we can see that as we move from the proximal part of the basin to the more distal part of the basin, we're getting enrichment in the iron content. So this line here is showing the iron concentration of the average Archean crust. And to the left of this line, we have mostly proximal samples, which means they are depleted in iron relative to the average crust. And on the right side, we see the more distal samples, and they are enriched in iron relative to the crust. You can also see that at the same time we have iron enrichment, we're getting increasingly negative delta 56 iron values. And this line here is representing the average delta 56 iron value of Archean crust. So above the line, we have mostly proximal samples, which means that they are enriched in the heavy isotope of iron. And then below this line, we have the more negative delta 56 samples, the distal depot facies. And that means that they are depleted in the heavy isotope relative to the crust. So this inverse correlation between the iron concentration and the iron isotope values has been seen before in modern environments. And it's indicative of something called a benthic iron shuttle, or a microbial iron shuttle. So this work was pioneered by Silke Severman, and in her 2008 study on the Black Sea, they found that isotopically light iron was being produced on the shelf by microbes, and this light iron was preferentially removed and deposited and trapped in the more distal deep basin. This meant that a residual heavy pool of iron was left on the shelf. Now, another important aspect of this study is that it required a redox boundary in order to trap the benthic iron flux, or the microbial iron flux. And in fact, every study that I've come across of a microbial iron shuttle requires a redox boundary in order to get that inverse correlation between iron concentration and iron isotopes. So this is a modern environment study. How does this apply to the Mesoarchean? And this is where it gets fun. So we've got a model. And the first part of the model is we need food for the dissimilatory iron-reducing bacteria. And so because other papers have come out suggesting that oxygen was present in this Mesoarchean atmosphere, I'm suggesting that there is a redox stratified water column with oxic surface waters on top and anoxic ferruginous waters below. So iron would precipitate and go to the shelf, and these ferric iron would be reduced by the DIR bacteria, and they produce isotopically light iron. Now, this isotopically light iron would be preferentially removed and trapped below the redox cline. And this would leave the heavy iron isotopes on the shelf. This is why we have the more heavy iron isotopes on the shelf, and they get increasingly light as we increase the distance from the shelf. So one of the remaining questions is, what minerals are the reservoirs for the benthic iron flux? I address this question by doing normative calculations based on bulk element data and iron speciation data. And so you can see that the green represents silicate as the iron-bearing phase, the red would indicate magnetite, the blue pyrite, and then the purple hematite. And as we move from the proximal to the more distal part of the basin, we're seeing an increasing proportion of magnetite. And this leads us to hypothesize that it's magnetite is the main reservoir for the benthic iron flux. You can also see that the size of the pie chart is increasing as we're moving to the distal basin, and that represents iron enrichment. But it's not just iron that's being enriched. This trend also holds for manganese. Manganese is increasing as we go from the proximal to the distal part of the basin. And another previous study on the banded iron formations of the starved shelf of these rocks found that manganese was anomalously enriched compared to all other banded iron formations of the Archean. So if we plot iron isotopes, against manganese concentrations, you can see that we have an inverse correlation here as well, where we have increasing manganese content, and that corresponds with more and more light iron isotope values. So this leads me to hypothesize that manganese is hitching a ride on the microbial iron shuttle. So manganese is being shuttled as well. So to tie everything together, we see an inverse correlation between the iron content and the iron isotopes, which is indicative of a microbial iron shuttle. And the inverse correlation between iron isotopes and manganese content is suggesting that manganese is being shuttled as well. The iron enrichment is corresponding to an increasing proportion of magnetite in the samples. And this study is particularly exciting because it is the oldest record of microbial iron shuttle that we have. And it shows that dissimilarity iron reduction left a basin-wide footprint 3 billion years ago. So this implies that there is an active iron cycling microbial community in the redox stratified ocean. And lastly, I think this emphasizes the importance of using basin-wide samples to address basin-wide research questions. Because if I had only sampled the proximal part of the basin, I would just see heavy iron isotope values and think maybe some iron was oxidized. If I only sampled the distal part of the basin, I would just see negative iron isotope values and think there's DIR there, and that would be the end of the story. It was only with the complete basin that I could be able to piece together a model of a Mesoarchean iron shuttle. So with that, are there any questions?
So you mentioned how you're moving the ferrous iron from the surface ocean into the deep ocean. So if the surface ocean is oxidized and the deep ocean is reduced, or anoxic, I guess, or dysoxic, depending on how you want to phrase it, how are you getting the ferrous iron through the oxygen and then immobilizing it in the deep ocean?
That's an excellent question. So there's multiple ways there's multiple different transport mechanisms that have been invoked in the literature for microbial iron shuttles. The first would be a ferrous iron traveling along the redox cline and then going down below. Another one would be the iron is transported by ligands, and that would be able to travel through the oxic surface waters that way. And the last is that the benthic iron flux is being transported as a solid, a nanoparticulate solid. And that is what I favor. But it requires that all of the benthic iron flux is oxidized if you're going to preserve that heavy, the light iron isotope signature.
Okay, any other questions? Alrighty.